Bom dia a todos. Muito bem-vindos ao primeiro webinar do programa BIOTA-FAPESP na comemoração dos seus 20 anos. É uma satisfação para nós todos estarmos realizando esse evento, organizando com muito carinho, com muita dedicação por parte dos pesquisadores, porque esta é uma parte importante de darmos um retorno à sociedade em termos do que o programa produziu, o impacto dele nos avanços do conhecimento científico, no avanço de governança, no avanço de aperfeiçoamento de políticas públicas e no avanço na formação de recursos humanos. É uma grande honra para nós estarmos recebendo todos vocês aqui neste primeiro webinar, um webinar que conta com a presença do professor Norbert Jürgens, da Universidade de Hamburgo, que fará uma palestra sobre o desenvolvimento de programas similares, mas de longo termo, de observação de biodiversidade na região sul da África, envolvendo a África do Sul, Namíbia, Botsuana e Angola. Durante o webinar, se vocês tiverem questões para serem encaminhadas, por favor, encaminhem para o e-mail contato@biota.org.br. Now I'm changing to English to the benefit of our invited speaker. So I was saying that it's a great pleasure for the Biota Program to start this series of webinars where we are going to present the evolution or the advances, the projects under the umbrella of the program produced in terms of scientific knowledge, public policies, and resources training. It will be a great honor to have Professor Norbert Jürgens as the invited speaker, and he'll present his talk just after Luciano, Verdade, and Simone Vieira, both from the Biota Steering Committee, present the summary of the advances of projects under this large area that is ecosystem, terrestrial ecosystem functioning, and landscape ecology. So it's also a pleasure for me to introduce Professor Luiz Eugênio Melo. Professor Luiz Eugênio is the new scientific director of FAPESP, and now he will address us in a welcome message. Professor Luiz Eugênio, please.
Thank you, Jolie. Good morning to you all, or good afternoon to Professor Jürgens, and to some of you who may not be in the same time zone as we are. It's a pleasure for FAPESP to start this series of webinars as part of the 20th anniversary of the Biota Program. The Biota Program is one of the best, if not the best, program that FAPESP has developed. It has been very successful, as Professor Jolie has pointed out. A number of aspects raised out of it, including not only scientific publication and the accumulation of knowledge, but also the influence of policies for the public sector and the partnerships with industries or service providers which are part of our society. For FAPESP, again, it's an ambitious program which foresee will have a long life. And with that, I conclude my introduction, just thanking all of the invited speakers, Professor Jürgens, Professor Verdade, and Professor Vieira, and most of all, Professor Jolie, for his very careful and good conduction of this program. Thank you.
Thank you, Professor Luiz Eugênio, Professor Jolie, for the words. And I will present some results from the Biota project. And, oops, here. Here. I only recently arrived to the steering committee of the Biota program, but I have a pleasure to take part of the Biota program since the beginning. And for me, it's a pleasure to open this series of webinar in celebration of the 20 years of Biota program. And for those who want to know more about the program, you can access our site. And the Biota program is a research program on biodiversity characterization, biodiversity conservation, biodiversity sustainable use, and biodiversity restoration. And since the beginning, during these 20 years, the Biota program had supported 281 research projects, including here thematic projects, regular research, and young investigator and São Paulo Excellence Chair projects. And in the area of landscape ecology, a functional terrestrial ecosystem, we have supported around 35 projects. In these projects, we have around 500 students that take part of these projects. And in terms of the divulgation of science, this project produces videos, books, textbooks, and more or less 1,000 papers with the results of this support received by FAPESP, BIOTA-FAPESP. And it was difficult for me to select the main achievements of these projects. And because of that, I will show a little bit some results that I can understand like the main results, but I can I'm sorry for in advance if I forgot something or if I present some wrong point from the projects. And I will start with one of the first projects supported by Biota program, that was this one that investigated the relationship between the land use and the human disturbance in watershed, and this relation with biodiversity and biogeochemical cycle. The main result of this project supported some political policy things in terms of depollution of the watershed in São Paulo states. And another results was identify the relationship between sugarcane burn emissions and the air quality. And this is the first evidence that the burn affect the air quality, and this can be related with the health of population living in near the areas where we have sugarcane burn. And in this supported the legal instrument that lead to prohibition of burn sugarcane. And in the end of the 90s, the pollution was a major concern, and Biota Program supported another initiative trying to propose biotechnology strategies applied to the contamination of soil polluted with heavy metal. And this was published in a book. And the Biota Program also is interesting in understand how the relationship between soil, microorganisms, and land use change, and supported a project that investigated the dynamics of the arbuscular mycorrhizal fungi and Araucaria angustifolia, one of the most important species in tropical forest in the south of Brazil. And the results of this project show that there is a big difference between the biodiversity of microorganisms in areas with Araucaria and in area without Araucaria. And another important initiative from the Biota Program was supported long-term monitoring initiatives. And we have now 3 initiatives in this way. One of that, the first one, was the Projeto Parcelas Permanentes, where was established 4 big plots in 4 different phytophysiognomies in São Paulo state. The principal one, like Cerrado and like submontane forest, Restinga forest, and seasonality forest. And another one was the gradiente funcional that established around 22 hectares in permanent plots in a gradient of altitudinal and environmental disturbance in northeast of the state of São Paulo. And now we have another initiative establishing new plots in northeast of the Brazil in Caatinga. And this initiative is important because we can publish, take part of the international initiatives in database, and publish some papers that with approach in questions in great scale and in time. And we have some example here, including an estimative of the number of tropical trees species in tropical, and we can have more than 50,000 tree species in tropics. Another one, more recent, discovered that the global abundance of tree palms in tropics, and that we have much more palms in neotropics than in the other areas. And this one that investigate the tree symbiosis and which control the decomposition and verify that the climate is the principal drive to the composition and that is related with tree symbiosis. And in terms of the microorganisms, we also identify the free living nitrogen fixer community in bamboo can input around 50 kilograms of nitrogen by hectare by year in tropical forest. And the another result showed that the bamboo overabundance alter the forest structure and dynamic in tropical forest. We have another project in course studying how the deforestation in Amazon affects soil microbial communities and how this can affect the emission of gas, greenhouse gas like methane gas. And another point was the relationship between the spatial heterogeneity and this specialization from the typical tropical trees, and this relation with the ecophysiology response of these plants to climate change. And we also have some results showing that the Restinga wood community has a homogeneous strategies to water resources acquisition, and this can have an important impact if you change the climate change, if you dry or the increasing temperature. And I want to point these three papers where we discovered that the foliar uptake of water fog by a tree species that occur in the São Paulo state, and this is very important because the leaves take water from fog, and if you have some change in climate that the fog decrease, we can affect this vegetation. And another result was we found that the carbon and nitrogen stocks is associated with the temperature, and that we have much more carbon storage in Atlantic forest than in the Amazon forest. And these carbon stocks can be affected by the fragmentation, and the edge effect has an important role in the carbon stocks and sequestration. And based on our recent knowledge about this forest, a group developed automatic monitoring to identify the age of the vegetation looking by two important species that occur in old growth forest and in forest in early successional stage. And another thing that I pointed as an important find by this project was that the conversion of the forest to pasture have consequences in terms of the temperature, the air temperature, the soil temperature, and this can change the nitrogen cycling and the greenhouse gas emission. And another one that presented the relationship between the land surface temperature and the fraction of woman modified areas in Atlantic forest. And they showed that if we can increase 4 degrees in temperature in areas without forest. And our results support 23 legal instruments in São Paulo state, not related with the landscape ecology or ecosystem ecology, but whole of the project supported by BIOTA-FAPESP program. And we have a lot of challenges in front of us, but I decided to point it here. This one is that the climate system is increasing drought and temperature stress in terrestrial ecosystem, and what tropical species will adapt to these new hypertropical climate changes, and how will this affect the ecosystem process and ecosystem service and the society in consequence. And thank you, and it all.
Thank you very much, Simone. And now we are going to have the presentation by Luciano Verdade. Luciano, it's with you.
Thank you. This way, I think I would cover most of the world audience that we may have. And let me share my screen to start my presentation. Okay, I hope you can see my screen. Well, it's a big pleasure and a big honor for me to be part of the Biota program, which I've been doing for the last 11 years with 3 projects, 2 of them thematic, and also as being a member of the steering committee. And well, I could say that behind these impressive numbers presented by Professor Simone Vieira in a brilliant presentation, there's a long-term learning process that has been facilitated by the Biota program. Of course, during all these years, we have been dealing with many complex questions, but even the most complex one, I think, could fit in simple questions that we try to answer during our lives. In other words, we've been trying to answer what, where, when, how, who, and why should we know our biodiversity. Let's begin to begin. And to understand our first question, what our Biota is, the Biota program has been granted scientific proposals in botany, zoology, microbiology, and genetics, for instance. We have a great number of plants and animals, both vertebrates and invertebrates, that have been described as part of the program, the Biota program, during all these years. And we've been also able to describe both diversity and variability, genetic diversity and genetic variability of many species that help us to understand what exactly our biota is. And in order to understand where our biota is, the Biota Program of the past has been granted proposals in the Atlantic forest, the Amazon forest, the Cerrado, which are maybe most of our, if not main, but maybe the larger biomes, we can say, in Brazil. But we've also been working in what's being called human altered environments, like urban areas and agricultural landscapes. And with agricultural landscapes, I mean lato sensu, including silviculture and livestock production. But also, in order to understand how our biota evolved and live, the Biota Program has granted scientific proposals at many areas or sub-areas of ecology and biochemistry, which help us to understand why we can find some species where they are, why some of the species are disappearing or some other areas and things like that. Well, keeping going on with these simple answers, in order to understand who impacts or is impacted by our biota, the program has been granting scientific proposals, for instance, on applied ecology, ecotoxicology, human dimensions, landscape ecology, and wildlife management. And of course, all these questions are becoming or become more complex when we introduce human dimensions. Let's say, for instance, ecotoxicology. We found, for instance, in one of my projects, the highest level of chromium and plumb ever described in a reptile in the common tortoise, freshwater tortoise, the Phrynis brioforanus, which is very common even in polluted rivers in Brazil, like Piracicaba River used to be, and one of its tributaries called Piracicamirim. We also found some levels dispersed in fish in the same river. So this is still a major problem that we are going to have to face in the future of our program. When we talk about human dimensions in all its complexity, well, it's virtually impossible to talk about biodiversity conservation if we don't consider the diverse human dimensions involved on it. Landscape ecology, in my opinion, is one of the best examples of interdisciplinarity because it really means a synthesis between two large areas like ecology and geography. And when we started to use a geographic information system, space changed from a theoretical dimension to a practical dimension. So we could see the species in real world. That was quite an advance for us. And of course, when we talk about wildlife management, we're saying about biological conservation of endangered species as well as sustainable use and conflicts or control of damaging species and monitoring them. So, but when we go to why exactly we should conserve our biota, the program has been granted proposals on biological conservation, like I just said, ecological economics, ecosystem services, sustainable use, and zoonosis. Of course, why? It's not a scientific decision. It's a decision that comes or should come from society. But in biological conservation, we try to understand why or should we conserve species in order to keep their ecosystems functioning. Why some species disappearance would imply in a great loss of economics or economic goods for human beings. Some of the species pollinate flowers of species that we eat, for instance, or use somewhere else. So why is also in the goals of the Biota program. And then we come to understand when all these topics of all these biotic patterns and processes change. And then the program has been granting studies on historical ecology on a sort of short-term basis, phylogenetics or phylogeography in a long-term basis, And when we try to predict the future, there's a lot of good projects dealing with the ecological modeling as well. But it's surprisingly, when is being the most difficult question to answer. When you consider, because in general, we smash time as a dimension. And for instance, we put our master's students or doctor students in the field, making surveys like 1 year or 2 year long in the field, and then we smash time and consider the results as a single photograph or a picture. But in general, there are processes that we no longer see when you do that. But when we consider time as a dimension, we realize that biota in ourselves live in a changing world where everything changes, including the biota and ourselves. So what? And well, I should clarify that I'm not quoting Bolsonaro when I say so what, okay? But when should frame our other questions, because we live in a changing world. And at this point, I would like to go back to one of the most interesting papers I've ever read from Frank Preston, published in 1960 in Ecology. And it explains the complexity of time as a dimension in terms of scales in a very simple and elegant way. He suggests that in short term, our results in this, ecological studies are influenced by sampling processes. Whereas in mid-term, ecological processes can be captured, and in long-term can capture evolutionary processes. In other words, if you're interested in surveying birds, for instance, if you go on the field in the night, you're only going to be able to detect night species. If you go in the morning, you're going to be able to detect day light species. But if you decide to go on the winter or on the spring, you're going to see different species as well. You're going to detect different species as well because of ecological processes like reproduction, like demographic processes that change from year to year in a scale that comes from months to year. But in a long term, if you're talking about decades or centuries or millennia, you can detect evolutionary processes that determine the patterns of distribution on a long-term basis. I think it's a very elegant way presented by Frank Preston about it. I always recommend to my friends, even being a paper, believe me, published before I was born, believe me. Well, and in order to capture from sampling to ecological and evolutionary processes concerning our biota, I would say, and I've already been saying that we need a long-term processing scale monitoring program. Of course, we believe we know why, but we still don't know what, where, and how to monitor our biota. And actually, we don't know either who should do it or neither when. So I would like to go to the final part of my presentation, reminding that in general, we focus on the diversity of biological patterns. But those patterns are determined by the complexity of biological processes, because biological processes mold the biological patterns that we measure in general. So we should, when we talk about monitoring by our biota, to understand it in a changing world, we should think about monitoring biological processes that mold biological patterns. We could call it biocomplexity when we talk about biological processes, or the complexity of biological processes that determines biodiversity, which would be the diversity of biological patterns. And I would say that the last part about it, and well, in that would be necessary, for instance, to talk about, for instance, restoration of patterns or reintroduction of species. In order to do so, we would need to think about recreation of biological processes. That would be important. And of course, in the future, as we are already doing, it would be fundamental to cooperate with other FAPESP programs and other financial agencies as well, in special the climate change program and the bioenergy, BioN project as well, because they deal with maybe the most important processes that result in land use change currently. So we need to see, for instance, agricultural production in order to generate bio-based fuels. But we also need to understand how climate change would affect and be affected by biodiversity. So I think it's fundamental to keep coordinated with these programs. So I would say that for the future, we should keep our simple questions like what we should do. And this is a conceptual question. Innovation would deal with how to do that. Governance would determine where, when, and who should do it. And of course, for society would be the place to say why. Thank you very much. I hope this is still in my time. Thank you.
Thank you, Luciano and Simone, for summarizing the results or the advances or the impact the Biota program had on the area of terrestrial ecosystem functioning and landscape ecology. And now it is a pleasure for me to introduce Professor Norbert Jürgens from the University of Hamburg in Germany. Professor Norbert will be talking about the regional SASCAL biodiversity observation network in Southern Africa. SASCAL is the short for Southern Africa Science Service Center for Climate Change and Adaptive Land Management. So please, Norbert, the microphone is with you.
Thank you very much. Yeah, good morning, everybody. I would like to start by thanking all of you for the honor to give such a presentation on behalf of your birthday. I already felt honored when the invitation came from Carlo Joly. I felt even more honored when listening to the progress report just given by Simone Vieira. Congratulations, more than 1,000 publications on such a wide spectrum of topics. That is quite an achievement. Yeah, I had been asked to give an input with a focus on monitoring, and that I'm able to do based on our African programs. I feel that Luciano Verdade just gave a framework, so I don't have to start with theoretical explanations, but I should start immediately by sharing the screen. And oops, here we are. And starting my presentation, which I compiled using PowerPoint. I hope this is now visible for everybody. The title is a regional SASCAL. I'll explain that acronym in a minute. Biodiversity Observation Network in Southern Africa, lessons learned. And I'll talk a little bit about the societal needs, the urgency, the feasibility, the output, and the context. I do it in a way that I start with 11 slides just to give you an idea how this SASCAL observation network looks like. And then we can go step by step onto various topics and follow this title, Lessons Learned. In other words, what would we do in a different way? What could we have done better, step by step? Okay, SASCAL and WASCAL are acronyms invented on climate conferences. We talk about regional science service centers, following the idea that the national scope is a problem in terms of environmental change because climate change doesn't know about national boundaries, etc. Therefore, at the Bali Climate Conference, a proposal was made to develop regional approaches. And luckily, with a little bit of discussion, the countries Angola, Zambia, Namibia, Botswana, and South Africa decided to cooperate with Germany, providing the bulk funding to develop such a regional science service center with a head office now based in Windhoek in Namibia, developing the thematic areas climate change, water, underground water, and river water, forestry, agriculture, and biodiversity, and their change due to increasing human land use. All these topics were developed. And we used this program, which I had the honor to coordinate from the German side, to also continue with another program that was already started 10 years before, which is on top of my list left-hand side, and which also has the title, the acronym BIOTA. This is by accident. It was not a plan to go along with BIOTA-FAPESP. Our BIOTA in Africa was having the meaning biodiversity transect analysis because we started with continental transects. And already in that program, we started monitoring sites in various countries. So that is a background we could use to establish, for example, within these 5 Southern African countries, 154 automatic weather stations, which run all the usual standard sensors. We try to meet the WMO standards. And all these 154 weather stations are open access, available for everybody. Here you see the website. And whenever you open that website, you will see all these weather stations with their present situation, temperature, rainfall, whatever. And if you click on any of these, then you get time series. For example, here I opened the month of the year 2019 as a temperature curve, the rainfalls, here's the humidity, and there are many more variables that you can see down here. You can also go to the daily list of a month or the hours of a day. All these data can be downloaded freely from the web. So they are really available 15 minutes after they've been measured and are really helping to fill gaps in especially Angola and Zambia, where there were only 2 or 3 weather stations before. In a way, our biodiversity observatories, and we have 60 now in these countries, are similar to the weather stations. They do not measure climate change, but change of biodiversity. And at present, we have 47 fully standardized square kilometer observatories. I'll explain what that is in a minute. And 13 more complex auxiliary observatories. And on top, there are several thousand single plots. The single plots are not revisited annually. However, all the observatories we try to visit each year after the rainy season. And also those data, like the WeatherNet data, are available on a website that's called observationnet.org. And before I show you that website, perhaps a short look at the design. All these biodiversity observatories are having a standard design. They are based on a square kilometer in the landscape, where 100 hectares are really marked in the landscape with metal poles, with number plates, so that orientation is no problem. And within each hectare, you have what you can see in blue here, that is 1 hectare. You have a center point, and north of the center point, there are vegetation sampling plots, 1,000 square meter, 100 square meter. I don't show the 10 square meter and the 1 square meter. And then south of the center plot, you can have a soil profile. And south of the center, there's an area for zoological sampling. Such a spatial subdivision makes sense to avoid artifacts. So everybody knows where to walk, how to get to a place, and what you are allowed to sample or not, following a number of rules in this nested multi-scale system. We normally look at plants in our Hamburg team, but in cooperation with other teams in Angola, Zambia, Namibia, Botswana, and South Africa. We look at the cover of the vegetation, but also at the abundance. So we also count individuals, what may sound funny, but it can be done in a nested system. We also follow up life histories of individuals. At the same time, there are drones used all the time, and we have automatic systems, and we measure lots of biophysical parameters. All these data are published here in environmental monitoring and assessment, a publication shown here. Now we go to those 60 biodiversity observatories, and the entry page looks like the weather stations grouped by countries or north-south, as you like. And I want to show you one of these. Let's take Subatsfontein, South Africa, down here. Then you have an entry page, observatory info sheet, that gives you general information on the coordinates. You can click the Google Earth ball to go to the spot using Google Earth. You can open a book where roughly 30 pages have been compiled as a PDF, giving you baseline data regarding geology, climate, soils, also what has been found with regard to microorganisms, biological surface crusts, lichens, higher plants, lots of different taxonomic groups of fauna. Or perhaps you want to have a compilation, integration of climatic data. Perhaps you want to have a look at the ranking, the 100 hectares within each square kilometer are always ranked with a random generator, making sure that all teams start at plot number 1 to have optimum overlap of all activities, because some teams, some disciplines are only able to do 1 to 10 plots with their workload, while others are able to do 20 or 30. You can also look at the soil data, downloadable, or the analytical data, remote sensing information, a photo guide showing all the plant species ever found on that plot. And now we started to also add the time series regarding plant diversity to start somewhere. And that means you have now roughly 20 years. And as you can see here, there are simple parameters like species richness. Often we exclude annual plants because they are just following the rainfall. It's more interesting to look at change in time due to land use or climate change. Also rainfall can be shown. We found quite helpful a more complex index which represents rainfall balance, following and integrating also the evapotranspiration of the last 12 months. And then you end up with a SPI index, which is either blue if the situation is really humid for plant growth, or red whenever you have a drought season. All this is presented openly on the internet. And we now also start to go even on species level. I'll show you a drastic, sad example. There was a dramatic drought in Southern Africa right now. And in the Richtersveld National Park in South Africa, here you can see, starting already in the 1990s, that the ups and downs of the usual rainfall, the gray columns, is mirrored by the plant species. And then during the last 5-6 years, there were rainfalls below the mean annual precipitation. And during that drought, meanwhile, since 2018, we have many plots, as you see them down there, right-hand side, where only mineral soil is left. Plots that in the past, looking left-hand side, had up to 70 species on 10 times 10 meter. So this is really the most drastic drought I've ever experienced in my life, and also the old farmers report that in their lifetime, nothing similar has happened. So we talk about climate change and the impact on biodiversity. Okay, this was my little introduction to make you aware how our program looks like. And now I've selected a few issues that may be interesting and may be stimulating or motivating. I'm very aware that the arid systems in South Africa do not really meet your beautiful Mata Atlantica situation. However, maybe these ideas are nevertheless inspiring. Okay, whatever you do, I have noted from your report right now that you are already full on board when I say what should be established should be a biodiversity observation system that is not only measuring what is interesting for scientists, but you definitely try to deliver products for decision makers. So you hand over knowledge that can be translated into decisions. So we did the same. We tried to have on board the political decision makers, ministers even, but also and mainly natural resource managers, those people who manage the water resources, the agriculture, but also conservation planning, the national parks, communities, etc. And most interested, the farmers. The farmers ask most questions in a week. They are really interested in the data, and it's not important whether they are commercial farmers or local communities. And there's the grassroots civil society, the NGOs. So the beginning of our project was really meeting decision makers, meeting stakeholders, getting in contact with, well, 50% of cabinets of countries. And after a while, we were successful and got important decision makers on board. Very important was that this lady, our Chancellor Angela Merkel, was happy to come to Africa for a full day to listen what the program is intending. And then we got her support. We are lucky to have a Chancellor who is a natural scientist. Okay, that put away. Now we talk about the design of an observatory network. Where do you put your observatories? Our decision was from the very beginning a continental transect line, which goes along the most important climatic gradients with the idea to cover the most important ecosystems in a zonal approach. These were the mega transects that we used from the beginning, and I immediately go to the south and show you that from the winter rainfall in the Cape through the arid Namib Desert, with the increasing summer rainfall towards northern Namibia, that was our start. At that time in 2000, we didn't dare to go into Angola due to the civil war with minefields. Meanwhile, the transect has been extended to the northern border of Angola. Yeah, within that transect, we put our observatories. And as I already showed a little bit, we had to take decisions about the sampling design of these observatories. I already showed you the 100 hectares within a square kilometer. We defined that the disciplines should always follow the sequence of priority given by a random generator and defined for all times at the beginning of the first year. And 20 replicates should be done. Even if there are different habitat types, those are distinguished, and then the random generator has 2 different series, and you move from habitat to describe the square kilometer correctly. Okay, that is the square kilometer. There's always an automatic weather station. The funnel here is a symbol for the automatic weather station. And on each hectare, we have the botanical and the zoological design I already explained. and their annual data are taken. That's how it looks in the landscape within the hectare. The whole picture, you can see the 1,000 square meter, the 100 square meter, the 10 square meter, the 1 square meter. And in such an arid landscape, it's easy to understand that we also count plant individuals, abundance, trees on 1,000 square meters, shrubs on 100 square meters, dwarf shrubs and grasses, etc., on 10 square meter. It's an approach that makes sense with 20 replicates on a square kilometer. And then you can be interested in many biological questions. Luciano Verdade just showed us that we definitely, as natural scientists, would like to understand processes and mechanisms in order to enable us to model and project processes. So when we look at such an observatory plot and we see trees of the species Aloe dichotoma, we may be interested, why are the tall plants having such a canopy which is broad, while the young plants look like a pole? And then you choose just one plant and you observe it each year, and that you can do with many. Then you see the baby and the growth through years. And only after 17, 18 years, you can see that the plant is creating branches and starts to flower. And now you can see some destruction at the stem. And the explanation for that strange behavior is this animal, the kudu. Because the kudus, with their 1 meter and longer horns, really kill the young plants when they are still able to reach the leaves. And only when the death zone of 3 meters have been passed rapidly, then these plants become a proper tree. Okay, it's an eye-opener, and only monitoring of individuals was enabling us to do that. But the more important approach is shown on this picture. These are 100 square meter standardized plots from the Cape of the Good Hope, right-hand side, then going through the Namib Desert, then going up the savanna summer rainfall in Namibia, going into Angola. And on the left-hand side, these more bluish columns are in West Africa. Here I put the margin of the Sahara over here, and here we go into the Sahel. And if you now look at the species number of plants in a 100 square meter plot, you can immediately see that the number of plant species in Southern Africa is generally far above the rainfall line, the blue line, while in West Africa it's below, expressing a much richer flora in Southern Africa than in Western Africa. So the comparison of data is easy when you have standardized observation. Maybe West Africa is poorer because of a 6,000 years longer suffering history from human land use that arrived in Southern Africa only about 2,000 years ago. Why do we look at abundance? Well, the vegetation cover is very important if you look at grazing. However, abundance gives more insight into processes, and therefore it's an effort we can recommend. And at the same time, we always analyze not annual rainfall and other climatic data, but we look at monthly or at least seasonal climate data. So that would be summer, spring, winter, etc. It's much more important than annual sums or means. Let me show you that with an example from a presentation short a while ago in July at the GeoBone conference given by Ute Schmiedl, looking at the Kalahari. Again, this is very dry for you in Brazil. However, there are different predictions made by colleagues who say this will become woodland due to the carbon dioxide fertilization of our atmosphere, while others say it will become desert and shifting dunes will take over. So what's happening here really by looking at seasonal rainfall curves and looking at all the major life form types and distinguishing them and counting individuals. If we make an analysis for the time series from 2005 until 2019, also having counts of all antelopes and livestock, then we can analyze and come to clear results that tell us that in this world, due to a decline in smaller game, which would eat mainly shrubs, there is an increase of the woody component, and the grasses due to an increase in large game shown here, which is mainly grazing. There's a decrease in grasses, and this is properly explaining an increase in the woody component of small shrubs. That would never have been possible based on cover value planning. Yeah, a very important warning is regarding the time scale of your planning. We talk about long-term observation. We know that climate is defined as an average of 30 years. Only then you do not talk about weather anymore, but climate. Change of biodiversity is not defined like that, but we need some patience. Let me start to explain that with a joke. Welwitschia mirabilis is a famous plant, the last survivor of a group of gnetales that was found in northeast Brazil with an age of 112 million years. And we found an old photograph from 1884, probably one of the first photographs ever taken in this part of the world. And we refound those plants, and they are still looking exactly the same, 120 years. Make you wonder whether monitoring is always necessary annually. Okay, forget about that joke, but the warning is you need core staff with permanent positions. You can't run an observation network only based on students, changing every 3 or 4 or 5 years, because then the taxonomic knowledge and the methodological approach causes problems. Yeah, while showing such historical picture archives, let me mention, these are also an extension of your timescale backwards. A nice example is a Racing Mountain next to a uranium mine in Namibia. Here's a Racing Mountain. Here you see a landscape. Why did I take this picture in 2005? Because in an archive, I found this one from 1917. The road is not yet there, but the bushes are already there. If you go back and forwards, you see there's little change. And if you go to details, you can identify each and every plant. And the small bushes are a fog-depending plant. The larger ones are an acacia, depending on summer rain. So the changes over 80 years are negligible. And at the same time, this means there hasn't been a drastic climate change. So to look back and to use photographs from historical archives may be a good idea to identify locations for an observatory, because now this is a plot recorded annually. Looking at the logistics, and you want to use automatic camera systems, automatic sensors, also vulnerable weather stations, it is very important to get along well with the local communities. To protect your equipment from vandalism as one aspect. However, para-ecologists that are trained to do the measurements, take photos, are making your program more feasible financially. You don't always need to send a scientist from somewhere into remote areas. Yeah, another recommendations, integrate experimental approaches, not within the observatory, that should remain untouched, but next to it. 2 examples, grazing exclosures and fire experiments. Here is a very simple, cheap experiment that you can put around a square kilometer with replicates. A nice little fence, 10 times 10 meter, shows you visible even from satellite image, how different the development would be if no larger mammals could graze, browse, and trample the landscape. Yeah, looking at fire, very important, also in Brazil. We started a program that we call purgatories, where, again, in a standard design, at each observatory in Angola, burning is done in different seasons to understand, again, the process and mechanism side of fires, but also looking at possibilities to use fire as an instrument to manipulate landscape in a sense of nature conservation. Okay, I come to the last point. Comparison of agricultural land use practices is very important, but now you see the color red behind data acquisition because this is a huge problem. We did several sites where we had different land use in a landscape and where we placed 2 observatories, 2 square kilometers next to each other. Here's an example. Along this fence line, 2 farmers took extremely different decisions about the future of their farm and their children, one could say. The one right-hand side overexploiting, the one left-hand side sustainable utilization with no overgrazing. And then we put our sites and we measure each and everything, and I'll show you just the abundance of mice and the species number of mice. And I add that those on right-hand side had no plants in their stomach, but only parts of beetles. So it's easily done, but the land use data are not easy to acquire because many farmers are shy to open their financial situation. So when you do that, make a good plan with the farmers and with farmers associations, etc., to be sure that you have robust land use data for your later analysis. Yeah, we all know that automatic data capture is now possible based on technological progress, saves a lot of money. Yeah, and I don't think I have to talk in detail about this. Everybody knows about that. We put that also on the internet. So there's an EVI tool we just developed that will be available for each and every spot in Southern Africa where you can go back 20 years and you can identify even single fire events that happened during the time and how they impacted on the green biomass. Yeah, I hope this will be the future of our observatories, more and more measuring systems. Already today, we have TDR moisture sensors in most of the sites, measuring the percolation of rainfalls into the soil. Yeah, obviously there are drones, obviously there are different trapping systems done partly every month, etc. I don't think I have to go to details. Everybody knows about that. And maybe that's already what I can tell you in 30 minutes. My summary is long-term monitoring is feasible. It is scientifically very, very interesting because it provides insight into the processes and mechanisms. The transfer of that knowledge is essential for conservation planning, essential for sustainable land use planning, and nowadays more than ever before, because it is unbelievable how quickly not only the climate is changing, but with increasing demographic number, especially the land use is changing, and I'm convinced the human pressure in terms of land use is the more critical pressure, not only in Africa. I thank you for your interest and your time. For further reading, there's a publication down there that you can download from the internet. Otherwise, yeah, we can continue this discussion on various means in the future after starting today with a blind approach to this presentation. Thank you for your attention.
Well, Norbert, thank you very much for the presentation. Surely there is a lot that we can learn and try to apply in our future projects within the Biota program. And I would like to start with a question that you mentioned the training you had with the para ecologists, and this started with Biota Africa, and I think it's still running. And how is the engagement of these young people when their turn as para, or their training as para taxonomists ends? I remember that one of them was a school teacher already, so was taking things from the field to the school. And so what is the average situation of them?
Yeah, that is a very important question. And I'm convinced that the para-ecologist program was one part of the success of the project. We did it in a very professional way. We advertised this position widely in local communities out there in the landscape. We got lots of applications, and we mainly selected young people who probably would never have seen a school in their life. If not, they would have got this job. Then they were brought on a initial training of 3 months. That was quite a challenge for many of our colleagues, because we had, during those 3 months, we had to ask everybody, the soil scientist, the specialist for bees, the lichen botanist, etc., to please invest 1 or 2 weeks and train on lichens and soils, etc. 3 months. And that was mainly done in a specific location. We started at the research station Gobabeb in Namibia, which is a very nice institute giving the right environment. And then training on the job continued over years. And every researcher who went there had to contact the para-ecologists before, because the other important role of the para-ecologist was they were mediators between the researchers and the local people, because there were lots of rumors what these strange people are doing on our postures. When the soil scientists started to dig their profiles and take samples, land grabbing got a new definition. No, I'm joking. But there are many fears, and local people, especially in parts of Africa, suffered in the historical past. So it was extremely important to have these young people on board, to also translate what these people are doing. Quite often other people were following the researcher, and also the results and the analysis of the results and the presentations included the para-ecologists. So we had the para-ecologists really on convention on biodiversity international congresses, and they spent there a week, and there was even a side event presenting the para-ecologists. So they were proud to be part of the project. They also translated the results to the local farmers and made it possible that many scientific results were then turned into application. So I can really recommend the para-ecologist program.
Okay, I have a question here from Fernando Maiello. He asked, in this observatory network, have you monitoring through metagenomic analysis similar to barcode? was any kind of data of this kind analyzed?
We did not any genomics or phylogenetics at large scale. We had a number of selected taxa where in the early days just AFLP data were collected and repeated, and later some microsatellites, especially on iconic species like Welwitschia. It's just in review right now. But we did not broadly embark on phylogenetics and genomics. No, this was not our scope.
Luciano has a question.
Yes, sir. No. I was just saying no questions. Sorry.
No.
No.
So how much of the results that are being obtained by this long-term observatory is being used for improving governance of the areas in terms of how much of this has been included in improving public policies in biodiversity conservation in those countries?
It's an enormous dimension, but not easy to measure. During many years, we were almost weekly invited to visit the weekly management meetings of many ministries. The African countries have ministries of agriculture, ministries of environment, with a tradition that each Monday morning, 10 o'clock, there's a 3 hours management meeting where any new topics are discussed. So we were really invited to feed into these meetings. This afternoon at 6 o'clock, I will be part in another Zoom conference discussing the feasibility and meaningfulness of large-scale biomass export from Namibia to Germany. Because Namibia asked for support. In Namibia, you have a strange process called bush encroachment, which is a permanent increase in the shrubby component. And we talk about shrubs of 3 to 5 meter height, which have thorns that long and which make it impossible to make use of that farm any longer if the bush is really thick. This is partly caused by overgrazing in the past, partly caused by the suppression of fire, and partly also caused by the fertilization of our atmosphere with more and more carbon dioxide. We know that woody components are increasing everywhere. And now the farmers remove that bush with hard work, with fire, etc. And they invest that much money that many farmers stop farming. And if that bush could be sold, it would be an enormous help. And now there's really the idea to export the annual bush from 30 million hectares to Germany. And it's really considered by the city of Hamburg to replace coal, oil, and gas in their energy production by Namibian bush. Can you imagine? Yeah, just one example that will happen this afternoon, this discussion. And yeah, like that, I could give you lots and lots of examples. It's quite astonishing how these ideas find their way into decision making. It's most important that you have many meetings, many workshops also in the countryside. I would recommend not only to have this focus on politicians in the capital, but to spread ideas and knowledge in the remote areas what is part of the field work of researchers at the same time. We really ask them and motivate them to talk to people when doing their field work and when sampling data. So it's a permanent discussion. I should add that the research done by the different disciplines is really relevant. For example, many soils in Southern Africa in the center are extremely nutrient poor. And the agricultural productivity is at 50% of the productivity per hectare in other continents. So hunger and farming problems are also addressed by the research, including soil scientists, including microbiologists that look at the mineralization of the organic matter, etc. There were even proper fertilization projects included, even using rhizobia, specialized for indigenous Fabaceae, which can stand the heat of Central Africa. So the whole spectrum from pure research in order to understand ecological processes and mechanisms, in order to enable models and projections, to applied research, even agricultural spread of methodologies was integrated in the project.
I have a final question for you, and then I have a question also for Simone and Luciano. My final question to you is how much the pandemic has changed the routine of the project. I know that for some years you lived more time in Africa than in Hamburg, but I think that now your movements has been restricted, and probably the whole team is more not in the ground looking at the things, but more concentrated in their own home.
I'm not quite sure whether I understood the question right. We have a special role as people from an industrialized country in the Northern Hemisphere, being part of a teamwork in Southern Africa. So luckily, all this project what I reported was really triggered by scientific networks. We did not start with a political program coming from the government, but it was networking among the colleagues in South Africa, Namibia, Zambia, Botswana, Angola, and Germany, which also brought me in a coordinating role, because I could apply for the money from the German ministry. But from the very beginning, we promised that this will only be a phase, and we will hand over the whole program to the African colleagues after an initial phase. And that has happened. So I handed over the coordination role already 3 or 4 years ago. There's now an African director, and if you Google zascal.org, you will find Dr. Jane Alwach, a lady running the program nowadays, with an African team, with a head office and a regional secretariat and an open access data center, an IT team at Windhoek, with national nodes in the countries. So there's a national node in Angola and Wambo, there's a national node in each country. That is still partly funded from Germany, but also partly from the African countries. And my own role at present is really shrunk to the biodiversity observation scheme, because the staff of SASCAL is mainly dealing with climate change and hydrology. They did not invest into positions for biodiversity specialists. So that is the remaining role. And therefore, I personally still live 6 months per year in in Windhoek is a wrong word, in the veld, somewhere working, doing field work. And the same is done by the team of my department at Hamburg, also those colleagues that you had seen in the little presentations from Kalahari and the fire network in Angola. They are also doing 3 to 6 months per year field work in Africa. And at the same time, we have many, many African students at Hamburg doing their PhD or their masters in our institute at Hamburg. It's a permanent exchange. Did that answer your question? You don't have the situation. You are Brazilians in Brazil. You can set up your program without such political complexities.
Yeah, I was wondering how much the pandemic of the coronavirus changed that.
Right now, yeah, unfortunately, very much so. I'm not allowed to enter each of these African countries. I was luckily at Windhoek when the president of Namibia announced that from now onwards, no more flights from or to Europe are allowed. And I really left my home 15 minutes later and got the last plane to Angola and from there to Amsterdam and then back home to Hamburg. At present, no field work is possible. However, with the para-ecologists living there, it's still possible to do the monitoring.
Yeah. Well, Norbert, thank you very much for your presentation. I think we've reached the time we have to close this part of our meeting. And I will ask all, I thank all that have been following. We had more than 200 people watching us through the YouTube transmission. And I think that it's time for us to think a little bit ahead with the calendar of the new webinars that we're going to have. The second one happens next week and is being focusing on the research that the Biota program has done on microorganisms. And we are going to have as invited speaker Professor Jona Mazet from the University of California, Davis, that's working on the Virome project. She's going to be talking about this program. And then in September, we have the one on bioprospection. And we are going to have as visiting speaker Professor Barry O'Keefe from the National Cancer Institute in US that's going to talk about how this research on natural products has been developing in US. So I invite you all to visit the FAPESP's website, where in the right hand, on the top, the right hand, you're going to have a link to see all the 8 webinars that are planned for this series in commemoration of the 20 years of the Biota program. I thank you all for following us this morning, and I hope to meet you next Wednesday discussing microorganisms. Thank you very much.